Good evening, sir. Shall I start? Sanjay sir, shall I start? Jai Hind, sir. Am I audible to you?
Yes, audible.
Okay, sir. So shall I start?
Yes.
Sir? Okay, thank you so much, sir, for your confirmation. So, good evening and Jai Hind to all of you, distinguished faculty members, speakers, today's speaker, researchers, students, participants joining us from different part of the country and across the globe. It gives me immense pleasure to welcome you all to this 10 days international training come workshop on advances and innovation in veterinary science for viable livestock practices. This international training program is being conducted by College of Veterinary Science, Animal Husbandry, Jalukie, Nagaland under esteemed Central Agriculture University, Imphal, in collaboration with Agriculture Forum for Technical Education of Farming Society and Vital Education Group, Rajasthan. This program is being conducted under the visionary leadership of Dr. Anupam Mishra, Honorable Vice Chancellor, Central University, Imphal, Chief Patron, Professor Indra, Director of Instruction, CAU Imphal, Chairperson, Professor Lalita, Director of Research, CAU Imphal, Convener, Dr. I. Shakuntala, Dean, College of Veterinary Science and Animal Husbandry, Dr. Amrit Gogoi, Convener, Dr. Dinesh Kumar, the Course Coordinator, Dr. Jitendra Mehta, President, AFTEFS, along with the dedicated efforts of organizing committee and faculty members of College of Veterinary Science and Animal Husbandry, Jalukie, Nagaland. This 10 days international training and workshop is being conducted on online platform Zoom, bringing together the experts and participants to share their knowledge and innovation in veterinary science and livestock practices. So, Jai Hind to all and very warm welcome to you. On behalf of the organizing committee, I would like to invite the today's speaker. Before inviting, I would like to introduce the speaker of today's technical session first. So our speaker for today's technical session first is Professor Sanjay Kumar Bodhakur. He is a distinguished academician and researcher who currently serves as a professor and head of the Department of Veterinary Parasitology at College of Veterinary Science, Animal Husbandry, Jalukie, Nagaland. With the profession career with profile academician record spanning more than 25 years, Professor Bodhakur has become a cornerstone on veterinary science and Northeast India, contributing extensively in teaching, research, and extension work. Before his current tenure, he dedicated 20 years of veterinary college in Aizawl, building a formaldehyde reputation. His expertise in parasitic disease, a widely published scholar with over of more than 100 scientific publications in national and international journals, he has been instrumental in bridging the gap between the complex laboratory research and practical application for farming community. Beyond his research excellency, he is a dedicated mentor who has successfully guided numerous postgraduate students, fostering the next generation of veterinary science and clinical studies. We are privileged to have him to share his deep-rooted knowledge and insight and decades the field level experience to us. Now, I would like to request to sir, please deliver his lecture.
Thank you very much. So, good evening to all the organizing secretaries and my dear participant. So, myself, Dr. S.K. Botakur, already my introduction has been given. So, let me start directly. My topic of presentation will be parasitic diseases, parasitic zoonosis in Northeast India. So, let me share the slides first. Okay, let's start. So, though the object of this online course mainly basically for livestock practices in India, but my presentation for today is limited to parasitic zoonosis, and main objective of this presentation to those dairy animal handler as well as livestock handler and persons who are related with livestock and other people who are directly or indirectly linked with this parasitic disease or linked with this first. Is it audible?
Yes, sir, you are clearly audible, but I request you kindly go to the slideshow mode.
Now is it visible?
Sir, slide is visible, but slideshow visible to us, but your voice is clear. Yes, Zoom is on.
Now,
It's on second slide, sir.
is it second?
Yes, sir, it's on second slide, but not on slideshow mode. I think Zoom will be okay. You can increase the Zoom size, but I think it's 67% is showing on our screen. Now it's on third slide. Now it's okay, sir. It's okay. It's okay for us. You can use F5 key for slideshow. Shortcut keys F5. F5, sir,
S5.
F5. Or on the menu slideshow on third, fourth one, one, two, three, four, fifth one, sorry, in sequence of, because it's on home. Slideshow.
Now is it visible?
Sir, slide is visible, but Zoom is over because the fonts are beyond the screen. Please resize and minimize. Slide is visible to us, sir. Okay. It's okay, sir. Now it's visible. No issue. Yes, sir. You can continue. We can read the content.
You can continue, sir. No problem.
No issue, sir. It's now clear to us.
Now
We
it's clear.
see
Now
the
it's clear.
content. You can continue.
Okay, let me start then. So we know that what are zoonosis? Zoonosis is a singular word, and if it is es, then it is a plural word. So zoon means animal and nosis means disease. So infection or agent that are naturally transmitted between human and animal is the zoonosis. And disease and infection which are naturally transmitted between vertebrate animal and human, who has defined, World Health Organization defines in 1959, this is zoonosis. Now, who transmits zoonosis? Say, if we say zoonosis, the human, one side, another is farm animal, wild animal, and vectors. So, farm animal include cattle, buffalo, sheep, goat, swine, and all, including dogs as pet animals. And human, again, one way it is involved, another way wild animals are involved, rat, mice, squirrel, raccoons, etc. And in between these, some vectors are involved. These vectors are like say they are transporting the organism, the zoonotic organism to their one host to another. These vectors may be mosquitoes, tick, lice, fleas, and many others insect. So, sorry, who are at risk in human? Population at higher risks, generally infant, children below 5 years, pregnant women, people undergoing chemotherapy, organ transplant, people with HIV or AIDS, and elderly people are they are more susceptible. And most susceptible other groups, that is, if we say in groups, the farmers, livestock owners, and the occupational group. So, because these groups, they share air or space with animal, and frequently they are coming in contact with domestic animal as well as with wild animal. So, who are at risk in animals? Some animal also equally risk. Suppose immunosuppression, high elder animals, some reptiles, sick, ducklings, puppies, animal with diarrhea, exotic and wild animals. Now, the zoonosis term coined by the Rudolf Virchow, who is the father of pathology, he said that between animal and human medicine, there is no dividing line, nor should there be. The object is different, but the experience gained constitute the basis of all medicine. So, keeping in this mind, we'll go ahead. And now let us see the roughly 68 million death occurred in 2021, if you see the human death, in 2021, 68 million people died. And the leading causes, top 10 causes account for 39 million death, or 57% of total death. Among these non-communicable disease, 43 million plus death annually occurs because of non-communicable diseases, maybe cardiovascular, cancer, or other respiratory diseases. Premature death is there, 18 million and more. And during those 2021 record, because of the COVID-19 impact, 8.7 million people died during 2021. And some record on suicide also there. That is also a remarkable record, 7,27,000 people. So let us see the parasitic zoonosis. Now let me switch to parasitic zoonosis. The main parasitic zoonosis, it includes arthropod-borne zoonosis, protozoan zoonosis, trematode, nematode, as well as cystode zoonosis. So the parasitic zoonosis impacting global health, if we see, the parasitic infection caused over 1 million to 2.5 million deaths annually worldwide. And among these all diseases, if we see, we know that diseases caused by many organisms, say bacteria, virus, fungus, and other parasitic diseases. And if we see the public health importance of these diseases, if we chronologically these are placed, then first comes to malaria. So it is caused by Plasmodium protozoa, and more than 850,000 deaths annually occurs, followed by schistosomiasis. Luckily, these diseases we are at least in Indian subcontinent, disease in human being is not recorded. Of course, in 1951, one record was there, but after that, no schistosomiasis recorded in human being is there. So followed by onchocercosis or other filial disease, and though they are causing 36 to 40 million people are estimated disfigured or incapacitated by this disease. This is a huge data that more than 36 to 40 million people are disfigured. And onchocercosis, luckily, this disease which causes river blindness, in India, this is not occur. But because of this filial disease, one terminology of the disability life year span, it is there in epidemiological studies that because of this, when a person is suffering with this filial disease or the other parasitic disease, their working capacity is less. Thereby, the most economic losses are there. That is the Delhi. And global impact, if we see that over 120 million people were affected by lymphatic filariasis. So that is a huge data. So the next disease is trypanosomiasis. And this trypanosomiasis, African sleeping sickness or Chagas disease, or the other some trypanosomiasis also occur. Particularly, this disease cause havoc in African countries, and another disease in American countries also it is there. But in Indian subcontinent, trypanosomiasis, which cause havoc in our cattle industry, or the mostly we know that camels are also suffering with this trypanosomiasis, particularly Trypanosoma evensi, and of late these are recorded in human being also. Again, another disease is there, leishmaniasis. Of course, this is not seen in livestock, only the pet animals are involved. This is also causing an widespread disease, particularly the northeast, and they have some Bihar and some parts of Jharkhand, it is there. And whatever diseases we have seen, this parasitic disease, these are the main cause of death to human being. And if we see these 5 or 6 diseases, these 5 diseases are the top 5 diseases, then the HIV or AIDS come, because these 5 diseases which are parasitic in origin, this is till that no any control measure is means effective. Why it is not effective? Because if we see the malaria, schistosoma, filaria, or trypanosomiasis, always between 2 hosts, it may be a human being or maybe an animal source, between 2 hosts, one vector is involved. So one way, if we go to suppose control the Plasmodium protozoa by treating the person or the suppose an animal who is suffering with some blood pathogen diseases, if we are going to treat the animal, and we have to see the control of vector also. That is in two-way control strategy should be there, and that is why this disease till that no 100% effective control measures has been taken. And the age, they are placing the age or the HIV in last part, because in near future we can expect that in near future a vaccine may be evolved or vaccine can be developed to control the HIV, like those COVID, the scientists could control within 1 year. So the other least important parasitic zoonotic diseases, which are soil transmitted, some helminths, say hookworm, ascaris, whipworms, toxoplasmosis. We know this is an protozoa causing diseases. It occurs ubiquitous in nature. It occurs almost in all livestock, and the definitive host is cat. And Giardia and Cryptospore disease is also there. These are also protozoan disease, commonly widespread protozoan intestinal infection they cause. This is in human as well as in animal. And least important is trichomoniasis. So let us see the first some nematode parasite. What are the nematode parasite? Because my presentation is based on zoonotic, so I will give stress on the canine species, because the canine is the main important source to get the infection by animal, by the human being. In human being, maybe a livestock handler, or maybe a dairy cattle, or maybe a group of people. So that is why I'm including this presentation here. So if we see the cattle, they are the source for some eyeworms, some stomach worms, Nephostoma spinocerebrum is there, skin disease, Sarcoptes scabia is there, which is zoonotic important. It occurs in other animal as well as human being. Large intestinal entomoeba histolytica is there, heartworm is there, Diarrhea imitis, and the small intestine, many parasites which are definitive hosts for canine species, say Dipeldium caninum, Toxocara canis, Ankylostoma caninum, Ancineria stenocephala. These are there in canine, but it can be seen in human being as well. So similarly, cattle, we know, sorry, in cat, the cat is the main source for the other species of animal as well as human being is the Toxoplasma gondii. It comes under protozoan disease. Similarly, a cat may be a source for others, say Nephostoma spinocerebrum, some small intestinal parasite, Dipeldium caninum, again, and heartworm disease also there. So let us see the Northeast map. So these are the 7 states of Northeast, right from Assam, Arunachal, Nagaland, Manipur, Mizoram. We know that these constitute the Northeast India, and now the Sikkim also, it comes under Northeast India. And this Northeast India, the vast mixing of people are there, their food habit, their cultural habit are different. So because this parasitic genesis, if we discuss, so food habit is also come, that I will come and cover all these things. So nematode zoonosis, only I will give, these are my personal works. This is not a review article. What are the personal works? I will give more stress. My experience, I spent more than 23 years in Aizawl, and most of the diseases which are important in zoonotic aspect, those were only included in this presentation. So dictyophyma, strongyloidosis, Nephostoma, dirophylosis, toxocariasis, ankylostomiasis, thalidosis, and dracunculosis. These are the main important nematode zoonosis. Now, first, let us know some. Nephostoma spinocerebrum is a nematode parasite. It's very rarely it also seen in pig, but this is an important zoonotic nematode, which is recorded in dog and cat in Mizoram, and findings were after that published. And mainly dog, cat, and other carnivore are involved in the life cycle. Cyclops and fish act as an intermediate host, and human being get the infection from the fish intermediate host. So these are some recorded of Nephostoma species. So this is the parasite. So whenever the nephostoma, their location in the stomach, during postmortem, we encountered some stomach lesions, and after that, when the stomach lesion were cut through, then some parasite were visible. After that, these were seen under light microscopy, then under scanning electron microscopy, and on the basis of egg findings, we confirmed that these worms were the Nephostoma spinocerebrum, and their histopathological studies were also done. So Nephostoma spinocerebrum was reported from cat, dog, and wild feline from different regions, including Northeast. So Borua and Gogoi, they have recorded, Deka and his co-workers, so myself and Dr. Mukherjee, Islam and co-workers, they have recorded. Additionally, this is important, that Nephostoma hispidum and Nephostoma dolorosi were also reported in domestic pigs from Northeast and Eastern region. Zadov and Tendon, they have reported in 1994. In the Northeast region of India, out of 6 human cases, so the previous things we described about, or we have recorded, this is in animal. Now let us see Nephostoma species in human being. In Northeast region, out of 6 human cases reported, 4 cases of nephostomosis due to Nephostoma spinocerebrum have been reported from Assam, mostly ocular clinical infestation. And some of the scientists, their name are also there. I'm not going to read out all those. And one is from the state of Meghalaya, Borua and co-workers. They have reported, another case reported was from Manipur. So Sharma and Bhattacharjee in 2022 reported first time a case of human nephostomosis caused by a third stage larvae of Nephostoma dolorosi in the right eye of a middle-aged non-tribal rural woman from Assam. Nephostoma dolorosi is a parasite of pig. And while studying this parasitic epidemiology, 4,520 freshwater fishes were dissected, were examined for the presence of larval helminth in Assam, and overall larval helminth was found 1,369 fishes, of which 21.4% and 1,348% number had L3 stage of Nephostoma spinocerebrum, and mostly Ophiocephalus punctatus, that fish harbored the larval stage 3, that means which is the infective stage, which is the infective stage for the human being as well as animal. It is transmitted by Ophiocephalus punctatus fish. Now, if we see the cat, we studied the cat parasite in thoroughly in Mizoram. There also we could record it Nephostoma spinocerebrum in 3 cats, and subsequently the paper was published in Asian Journal of Tropical Health, Public Health, long back. So these are some pictures I want to show. This is a cat intestine. So this is the cystod parasite, Teniformis, and some other nematode parasite also seen. These are Toxocara cati, and this is a zoonotic parasite, the Physocephalus, Physoloptera. This is seen in cat stomach. So next important zoonotic nematode parasite, if we say this is the Diarrhea-Philiasis, this is an heartworm of dog, but it also occur in human being. Almost all warm-blooded animal, they may carry this parasite, and mosquito are the important, or mosquito, more than 56 different species of mosquito are responsible for transmission of this parasite from animal to human being, or between or among the animals. And we could record it. An elaborate study has been made in Northeast India on occurrence of heartworm disease. We established endemicity of heartworm disease in any region. Average 18.03% infection rate was recorded. Epidemiological study was conducting utilizing conventional serological and molecular approach, and the study concluded that not technique is the gold standard technique, and it will be give other immunological test. If we conduct, it will give the 100% accuracy of those tests. So similarly, we could also record another filial parasite, Diarrhea repens, which is coexist or overlap with Diarrhea imitis in any state. And dog act as the reservoir host or source of human diarrhea. So we could record more than 10 papers on Diarrhea imitis in animal as well as in human being also. So cases of human diarrhea, now if we see, are increasingly being reported in last few decades all over the world and can be considered as emerging zoonosis. Most of the cases reported from India are ocular diarrhea, where the worm is detected most commonly in subconjunctival or periorbital tissues. 3 subcutaneous cases reported from Dibrugarh in Upper Assam and another district from Assam, Eastern Assam, within a period of 6 months in 2011. And 3 cases of human ocular diarrhea also reported in another district of Upper Assam. And this is recorded in subconjunctival sec and reported by Nath and his co-workers. 5 cases of unpublished ocular diarrhea also recorded from Sarkardev Netralaya in Guwahati, reported by Das and co-workers, and other nematode parasites. So let us see other nematode parasite, toxocariasis, which is caused by Toxocora canis and Toxocora cati, are ubiquitous, and these are frequently encountered in dogs and cats. And these parasites are mostly responsible for visceral larva migrants in human beings. So though these parasites are there, adult parasites are found in dog or cat, but this larval stage, if these are infected to a human being, particularly the young children, they may suffer with visceral larva migrants. These cases, these were reported through her studies on public health importance. So she revealed that more than 30% prevalence of ascarid in dog population in Assam and 12 potential zoonotic other GI parasite also she could record it from Assam. So other least important nematode which are reported from different parts of India are in these slides. This is depicted some of the parasite occurrence of toxocariasis, Toxocora canis from Delhi, some from Kashmir, and some are reported from the other parts, say from Kerala. In Haryana also it is reported that 6.4% out of 94% to be seropositive of Toxocora canis. So Traub and co-workers, they have reported that 93% prevalence of dog hookworm in Assam. Molecular study of dog feces revealed 72% of dog will harbored with Ankylostoma caninum. This is a hookworm disease where the dog is the source, but the human may get the infection through soil. Suppose a milkman is engaging with the milking of a cow, he may get, if he is barefooted, then he might pick up the infection from the soil, because in case of hookworms, the larval stage 3 is they are reached to their definitive host through the skin penetration. So similarly, Lindman and co-workers conducted hookworm infection among the rural tribal population of Meghalaya, where they could detected Nicator americana, another hookworm diseases in human being, where the source is an animal. So these are the global pictures of hookworm infection in Indian scenario. 1027 population. It was recorded 1027 million population, 534 million are rigs, and infective prevalence was recorded 7%. So this is the pictures of Indian subcontinent. And another parasite, dictyophymosis. This is Dictyophyma renale. This is a giant kidney worm of dog. Though we could not record from Northeast India, but it is recorded from other parts of country. Sohan and co-worker, they have recorded from Meerut. Venkatesh Raja, they have reported from South India. and some review of India. These two reported is recorded. So another important nematode parasite is the eyeworms, where the vectors are some flies are involved, Thalesia calipedia. This is the first reported from Pakistan in 1910. So they caused canine ocular thalasiasis. And in Northeast India, this is reported in Nagaland. Recently, we could report it from Jalukie, these parasites. And the human thalasiasis in Northeast India recorded, many cases were recorded from Manipur and Assam. And similarly, from other parts of country, Tamil Nadu, Himachal Pradesh, Karnataka, Chennai, it is reported, but many cases are reported from Northeast India. So in 2018, one case was reported from another district of Assam, that is Morrigaon district of Assam, in human being, where the source may be an animal. So another important, some livestock parasites, strongyloidiasis, they are strongyloidiasis or threadworm infection, particularly strongyloides starcoralis and strongyloides fuelworni. They cause skin infection in human being. Also, these are the origin in livestock. So this is the highest number of cases was reported in Delhi. Out of these 36 cases, 30 cases have been reported from a territorial level referral hospital. Next in order in Assam, 17 cases, Maharashtra 17, Karnataka 16. So that means these were prevalent throughout the country. So now let us see the cystode parasites. So some of the canine origin cystode parasites are, one of the very important cystode parasites is Dipylium caninum. In Northeast India, Devi and co-workers, they have reported occurrence in canine species, but Traub and Arze also she could recorded this occurrence in dog from Tezpur in Assam. And these are the some figures from other part of the country. And if we see the occurrence in human being, of course, there is no any recorded cases occurrence of human being is seen in case of Dipylium caninum, but this parasite is transmitted by a flea. So maybe some human being are also there, but the recorded cases are not found in the literature. So another parasites, echinococcosis, we know that the hydatid disease. So hydatid disease, the worm responsible for causing or in the life cycle of this parasite, one stage is hydatid cyst, and the mature or the adult parasite is Echinococcus granulosus, and like that 4 species are there. This is a parasite of canine or particularly dog, but this hydatidiasis or hydatidiasis may be occurred almost all warm-blooded animal. Even most commonly it is seen in sheep and goat, buffalo, cattle. In meat inspection or in slaughtering or in post-mortem, the encountering of hydatidiasis is most common. So these are the some cases cystic echinococcosis recorded in different parts of India. So I am not going through because otherwise it will take a lot of time. So many cases are recorded. So geographical areas, Chandigarh and Central and South Asia, South Andhra Pradesh, West Bengal, Maharashtra. That means many parts of the country, the hydatid cysts are recorded. So Borua, similarly, if we see in Northeast India, Borua and co-workers, they reviewed and clinical laboratory features of patient in human being, and they reported some cases in human being which were suffered with hydatid cyst. The most common site of involvement was in the liver, followed by lungs and brain. And similarly, in Shankardeva Netralaya in Guwahati, 3 cases of orbital hydatidiasis also recorded, and this is reported by Das and co-workers in Eye and Brain Journal. So between October 2008 to September 2013, 37 patients were operated in the Department of Cardiothoracic Surgery in Guwahati Medical College in Assam for lung parenchymal hydatid cyst. 26 patients were female and 11 were male. The mean age of the patients were 30.6 years with a range of 16 to 44 years. So the source is a small tenid eggs which cannot be seen through our naked eyes. So any livestock owner, any persons, somehow if they get the infection through ingestion of these eggs of Echinococcus species, he or she may suffer with this hydatid disease condition, and the water or the other source are the main source, water or other feed are the main source for getting hydatid infection by human beings. So this is an example. Of course, these pictures, these are some pictures because the Echinococcus granulosus, these are the parasite of dog. So my intention is to show these pictures. So I visited some of the dog where dog are killed. So some persons they consume dog meat. If we see this person who is putting in cigarette, he is smoking, and in one hand, this is a bare hand, he is cutting some the dog intestine, he is handling dog intestine and this, and there is every chance of that this person, this butcher, he has the chance of getting hydatid diseases. So let us see the other parasites, other cystoid parasites, sparganosis, which occur in human beings, is caused by Spermata mentionoides. It also some toad and others snakes are intermediate hosts. In Northeast India, this is also reported. Similarly, in human being, other part of the country, also many reported cases are there in human being. And another the important parasite of cystoid parasite is Diphyllobothrium. This is origin a broad fish tapeworm, where the fish is the second intermediate host and canines is the definitive host, and the human being may get the infection through ingestion of this infected fish. Infected fish, this is a life cycle. I am not going to deal all this to say details about the life cycle of these parasites. But the human cases, if we see the deviant cohorts, they have reported human phyllobothriasis from Pondicherry, and Pancharatam co-workers, they have reported. This is from Vellore in South India. Ramanna and co-workers also reported from South India. This is occurring in human being. So beside this cystoid parasite of canine origin, we know that some of the other cystoid parasite where the cattle, Tenia segineta, is an important cystoid parasite where the human are acting as intermediate host for this Tenia segineta. Similarly, another pork tapeworm where the pig act as an intermediate host for Tenia solium. Tenia solium is a cystoid parasite which occurs in human being, but the pig act as an intermediate host. So from this livestock, pig may be source for human being for Tenia solium, and Tenia segineta from beef, a human may get the infection. And in this case, the public health importance, and in this case, the one has to be aware about the taking of beef and pork. So we must at least make awareness of these cystodes occurring in human being, and particularly during the meat inspection. In pig muscles, these are commonly found, as well as in beef, these are commonly encountered cystoid parasite. Now let us see some trematode parasites. So in Northeast India, the paragonimesis is a very important zoonotic disease under trematode parasites. The Paragonimus westermani, this is a lung trematode. This basically this parasite is seen in canine species, but in Northeast India, many human cases are recorded. 22 million people are worldwide infected with this parasite. In India, Paragonimus was first reported from Manipur from the Regional Institute of Medical Science. Singh and co-workers, they have recorded in human beings, subsequently described from Arunachal Pradesh and it is reported by Regional Medical Research Center from Dibrugarh in Assam. And paragonimus and paragonimesis, a review article is there. Suppose some are interested, they may go through this review article, paragonimus and paragonimesis, published in Indian Journal of Medical Research. And in dog and cat, we could also record this parasite from Mizoram, and in other part of Indian other state, Rao he could record it, and Srivastava also recorded this parasite in animal. So the presenting author reported 2 cases of paragonimus in dog in Aizawl, and possible road to human being also speculated. So let me go quickly some slides, because this parasite, in the life cycle of this parasite, if we say the zoonotic parasite, how they transmitted from animal to human being, there is always involvement of some vectors or some other intermediate hosts are there. So how a human may get the infection? If we see that epidemiological point of view, so these pictures will be more clear. So in this case, we could recorded 2 cases. In slaughterhouse visit, we recorded 2 cases of dog harboring paragonimus infection. Number of dogs screened were 100, and it was diagnosed on the basis of worm morphology and egg morphology. So let us see, this is a speculation. We could record this paragonimus species. It is the intermediate host. Second intermediate host is the crab. So most of the state in Northeast, the people, they are fond of taking crabs in their table. So the crabs are eaten, and one preparation is made particularly in Mizoram, and they call it as ayum. So it is their own language. In ayum preparation, they are using raw crabs. So let me show some pictures. So these crabs are ready for preparation of ayum, and these are the some sesame seeds, and now they are mixing. This lady is ready to mix these both the crab and sesame seeds. So they both crab and oil seed are ready, and this is how she is mixing. First oil seed are grinded, then they are mixing with raw crabs. Now these raw crabs are mixing with these crabs, and some part of these crabs, some offals we can say, they are thrown to this dog. They are rearing dogs also, and later on, these dogs, when we sacrifice, and we could find these paragonimus species. That means, in that case, we speculated that source were those crabs. And these dogs are also fond of eating these crabs. And this is the ayum preparation, and they are keeping in some bamboo-made devices, and they are taking as fermented, and after that they used to take. Similarly, in some places of Northeast India, the snails are marketed. The snails in Mizoram, they call it senkhal. These are marketed. And the snails, some people, they are fond of taking snails. There is no any hindrance, or there is no any objection, the food habit of peoples. Because different people, they have the different habits. But the main thing is that they are unaware that snail may act as a source for many parasitic zoonotic diseases. And here the lady is selling the snail, and in one side she is selling some orange also with this bare hand. So the chances of getting infection with the Sarcaria, because the Sarcaria is one stage of infective stage of some trematode parasites. By the contaminated hand, this seller may get the infection. She may pick up the infection from those snails. And if the snails are taken, this person, he is preparing the snails. How he is going to take the snail? These are the snails, just putting in the warm water, and he is taking the snails. And these are some common pictures. These are some food habit of Northeast, some tribal peoples in hilly regions. So there are many chances of getting infection from these crabs by those people, because these people. So someone may ask in livestock section why these are shown, but that person, he or she may rear the many other livestock, because almost in all tribal population in Mizoram, they are keeping pig in their backyard along with the poultry. So these are the preparation of crabs, preparation of frying of crabs, but some raw crabs are also keeping. So this contamination, every possibility of contamination of human being may be there. So these are some pit also. And these paragonymous reported in human being. I already uh, elaborate these things in Manipur. This was recorded. Similarly, in Aizawl also recorded in human being. In Assam also it is reported. Similarly, in Nagaland also these cases were reported. But the most important thing for our participant, suppose some maybe not from the parasitic field. So why this is important? This paragonymous, when the lung infection is there in human being, the disease is mimicking with TB, tuberculosis. And most often the symptoms are similar with TB, and this is oversighted. Whenever some is approaching the human medicine, they may think that the people, he or she is suffering with TB, but in fact, he or she is suffering with paragonymous. So that is why the disease is very, very important in human or public health aspect. Some other parasitic disease, some other uh, important or least, we can say these are important parasite for the livestock, but these are zoologically least important is Fasciola species. But it was reported from Assam, Fasciola hepatica. This is an important trematode parasite of livestock, but a 7-year-old girl case was recorded in Assam, and this is the first human cases. It was reported from Assam, and it was published in Journal of Communicable Disease in 1997. Another important trematode is Fasciolosis baski. This is a parasite of pig, but it is also occurred in human being. A fluke of pig has been reported in human being from Assam, and this parasite may be picked up because of some food habits, because of this metacercaria. These are the life cycle. If you go the life cycle of this parasite, the metacercaria or the infective stage of these parasites are seen in some water caltrop and other edible things which are grown in the water. So that are the source for this parasite. But originally, or the definitive host for this parasite is pig. Similarly, the heterophyte, one another least important trematode parasite, it was also reported from Assam and reported by Mahanta and co-workers in 1995. So let us see some arthropod parasites. So Sarcoptes scabiei, it occurs in almost all livestock as well as in canine species. This is transmissible between human and other animals. So 13 to 59% rural population has been reported this parasite in India. And among other arthropod zoonosis, we could detect Coxiella burnetii and Borrelia burgdorferi ticks from goat in northeastern state of India through molecular survey, and it was published in International Journal of Ecology from Mizoram. So these ticks are harboring in goat, but the spirochetes, these are Coxiella burnetii and Borrelia. These are the disease-causing agents which are transmitted by the ticks. Similarly, another important tick-borne disease, scrub typhus, also reported from Mizoram, and this was reported in 283 positive cases. Seroepidemiological study was done. 4,081 sera was tested from October 2014 to December 2016, and we could record these parasites, and this paper was subsequently published. So now let us see some protozoan diseases. Important are toxoplasmosis, trypanosomiasis, cryptosporia, giardia, and leishmaniasis. Trypanosomiasis, in beginning, I just said that Trypanosoma evensii, which cause sura, and this sura in livestock, almost all livestock are recorded from India, and in but few cases in human being are recorded. One recorded case from Chandrapur in Nagpur, Joshi and co-workers, in West Bengal, Sengupta and co-workers, and in Bharodia and co-workers, they have reported again from Gujarat. That means some cases of Trypanosoma evensii, it is reported from India in human beings, where the source is livestock. These causes are human caused by the Leishmania pneumoniae. The source maybe some are reported in dog from India, Ahuja and co-workers, Sharma and co-workers. They have reported from India, where the dog may be the definitive host. And let us see another parasite, Toxoplasma. This Toxoplasma gondii, this is an almost in all warm-blooded animals, and this is the most important pathogen, public health important disease. So cat is the main source, but almost all human being or almost all animal are suffering with this Toxoplasma gondii. So they cause early abortion, particularly in pregnant women, and as well as some deformed birth is also there, particularly the congenital toxoplasmosis also it's occurring. So these are the maps showing compared seroprevalence and incidence of rate in various parts from India. So giardiasis, another we know that giardia, they call giardia intestinalis, giardiosis, synonym duodenalis, giardia duodenalis is a synonym, or lamlia. So they cause giardiasis or the traveler's diarrhea. So it occurs almost all animal as well as in human being, where the animal may get the animal may act as a source for human being. So in genotypic study, it recorded A, B, C, D, E, F, Z, up to 8 different assemblance are there, where B and A are the most important assemblance. This occurs human as well as in animal being. So in India, both A and B assemblage of the Giardia duodenalis is recorded. It is first reported by Traub and co-workers in 2004 from Assam, and Suleiman and recorded from Hyderabad. So these cases were recorded where the both livestock, canine, and human are shared by this organism. So Cryptosporidium, another pathogen disease, Cryptosporidium parvum is the causative agent. So these parasites are also occur in almost all animals. And this study was, we made studies in Mizoram. So the Cryptosporidium in cattle and in human in Aizawl district, it was carried out. A total of 200 sample, 100 from cattle and 100 from human stool sample were collected, and it was detected Cryptosporidium oocysts by modified gel-Nielsen staining techniques after concentrating oocysts by Cetar sucrose, and we could record it. And this was a 100% definitive diagnosis was made and molecular method, and we could record it difference of Cryptosporidium were 13% in cattle and 5% in human being. So these were journal in 2000. These are the so as a veterinarian, our part is to see whether this Cryptosporidium is in animal. So this is the record from the medicos. So these are the geographical distribution of Cryptosporidiosis in India. So other zoonotic important protozoa, we could record it, Babesia microti, because in livestock, Babesia is a important hemopathogen disease, but there are very few cases are reported from outside of India that Babesia of cattle origin occurs in human being. But one important Babesia species, Babesia microti in India, is an emerging tick-borne zoonotic parasite. So it was recorded 8.8% occurrence in rat in northeast India. So this was done molecular detection of Babesia microti in laboratory mice we could record it, and it was published in Journal of Vector-Borne Disease from Mizoram. So these are the some reported, and we could also conducted some assessment of zoonotic helminth contamination of environment in Aizawl, and where the vegetables, fruit, and fruits and soil were evaluated for presence of zoonotic parasitic egg, and we could record it this parasitic disease in among school children below the age of 10. About 1,000 soil samples were screened, and 183 samples were positive for zoonotic helminth, and these were Ascaris, Ankylostoma, these trichoris or Ascaris, Ankylostoma, these Toxocara livestock also. 123 vegetable sample from different market area were assessed, and 164 vegetables we could found positive for many parasitic larvae. A total of 110 human stools were examined, 4 primary schools we aimed, and 27 samples were positive for helminthic infection, where sources may be an animal being. And in conclusion, the parasitic zoonotic constitutes some of the most important and common infection threatening human population in India, as well as in other countries, as well as in Northeast India also. The major disease associated with companion pet animals is described. With the status of infection in man and animals, we can afford to highlight gap in knowledge and potential intervention to prevent or limit the spread. Of course, some of the zoonotic parasites which I included or included in this presentation, most of the zoonotic parasites, though it is there, the source may be canine, but many are there where the source. Slide. Thank you very much for your patient hearing.
Thank you so much, sir, for
Thank you, sir.
such an information. Thank you so much. We are moving forward for next session. Thank you so much, sir, for enlightening us.
Thank you very much. Thank you very much.
Thank you, sir. Thank you.
All right.
So moving forward to the technical session 2, I would like to introduce our second speaker, Dr. N. Umakanta. Sir is presently working as a senior scientist in agriculture biotechnology in the Institute ICR Research Complex for Northeast Hill Region, Manipur Center, Manipur. He is an author and co-author of more than 75 peer-reviewed research articles in national and international reputed journals, and more than 3 books, 16 book chapters, and many conferences. He has attended many conferences and he has written many popular articles. Dr. Umakanta is the recipient of Early Career Research Award in 2018 with a research grant of 3 years by the Department of Science and Technology, Government of India. Presently, he is actively involved in the research area of genetics improvement of black rice development and understanding of the molecular mechanism of biotic and abiotic stress tolerance in northeast rice through molecular breeding and improvement of rice genetics through CRISPR and Cas9 technology, diploid haploid technology, and micropropagation of local banana. As a nominee of Government of Manipur through the Department of Horticulture and Soil Science, Dr. Umakanta acquired his BSc degree in horticulture from College of Horticulture and Forestry, Central Agriculture University, Arunachal Pradesh, with the University Gold Medal in 2005 from Central University of Imphal, Manipur. He is All India topper of the ICR JRF in 2006 for Plant Biotechnology Group conducted by ICAR New Delhi. He received both master's and PhD in the discipline of molecular biology and biotechnology from ICR Indian Agriculture Research Institute, New Delhi. So now I would like to invite Dr. Uma Kanta to please deliver his lecture note.
Thank you, Nisha Rana. So without wasting time, I will start my presentation. So I am sharing the slide. Uh, is it visible, no?
Yes, sir. Perfectly.
Yes, sir.
Okay, thank you, thank you. So I'll start the presentation. The topic is the enhancement of the nutrition and the health of livestock through plant biotechnological tool. So as a introduction, as it is already known to you that the Indian livestock population is around 536.76 millions. This is based on the livestock census 2019. Around 95.78% of the livestock is in rural, and well 4.22% is in urban, which act as a crucial pillar for the rural livelihood and contribute roughly around 4.11% to the national GDP. And livestock also contribute about 25.6% of the total agricultural GDP. And interestingly, India ranked first in the global milk production, third in the egg production, and fourth in the meat productions. In total bovine is 303.76 millions, which include the cattle, buffalo, mutton, and the yak. And the cattle population is around 193.46 millions. Goat population is 148.88 millions. Buffalo population is 109.85 millions. Sheep population is 74.26 millions, and other animals include camel, pig, and horse, pony, etc. So poultry, on the other hand, poultry is around 851.81 millions. Among the states, Uttar Pradesh has the largest livestock population, followed by the Rajasthan and the MP. So in India, the major livestock is the cattle, which is the largest cattle population with indigenous breed like Gir, Sahiwal, and Cancres. And the buffalo also rank first in the world with the breed like Mura and the Jafrabadi contributing significantly to the dairy sector. Goat and the sheep is the key rural livelihood and also known as the poor man's cow. And the poultry also rapidly growing sector supporting the economic developments. However, the challenges livestock in India is the low productivity. Generally, low productivity compared to the international standard, it is low. Another one is the resource constraints. There is a not available for adequate feed, fodder, and veterinary services. And also there is a high risk of health due to the frequent outbreak of food and mouth diseases, which causes the low productivity. So, low nutrition in the livestock of India is a critical challenges. Over 70% of the animals suffer from the mineral deficiency and the severe fodder deficit. A 65% shortfall in the green fodder and 25% in the dry fodder, where with the over reliance on the low quality crop residue lead to the poor productivity and the low fertility and stunted growth. This crisis is exacerbated by the dwelling grazing land and the climate changes affecting the feed availability. So the key causes of the livestock nutrition in India may be listed as feed and fodder deficit. India faces a major gap between the demand and supply of the nutrient-rich fodder, which is around 65% shortfall in the green fodder. And over reliance on the crop residue. Most livestock are fed low quality agricultural residue like paddy, wheat, straw, which are deficient in essential protein and nutrients, and a lack of pasture land due to the urbanizations, degradation of the grazing lands. There is a huge amount of decreasing in the area of the pasture. So there is a reduction of the natural fodder availability. Nutrient deficiency. Nearly 70% of the livestock of India suffer from the imbalanced nutrient like the calcium, phosphorus, and essential vitamin like A, D, and E. And climate change, as we all know that there is a climate change. So it causes the drought, heat wave, and erratic rainfalls, heavily reducing the crop yield and lower the nutrition quality of the fodder crop. And low adoption of technology, limited use of the scientific feeding procedures such as urea treatments of the straw and tailor-fit rations. So because of this factor, this this there is a huge impact on the livestock and the farmers. Indian cattle production is significantly less compared to the global due to the malnutrition. And another one is the poor reproductive health, and so low income to the farmer. So one of the measure may be the intervention of the plant biotechnological tool for the livestock. As you know, the plant biotechnology tools are revolutionizing the livestock management by enhancing the nutritional value of feed, improving digestibility, reducing anti-nutritional factor, and strengthening the animal health through the modified forage and the grain crop. Key application of the biotechnology tool may be crop improvements, the creating drought, pest, and herbicide-resistant crop, enhancing the production of the green fodder, and promoting the high-yield nutritious forest crop. Second one will be the quality improvements, enhancing the nutritional value and extending the shelf life. Third one may be the conservation, using technique like cryopreservation to save medicine and endangered plant. Fourth one may be the molecular farming, utilizing plants to produce useful compounds such as vaccine or pharmaceuticals, which will be useful for the livestock. So, yes, for the improvement of the livestock, I have listed 5 biotechnological tools. First one is genetic engineering, also known as a recombinant DNA technology, which involve the inserting desirable genes from one organism to the another, resulting in transgenic plant with the improved agricultural traits like the BT cottons, BT soybean, BT maize, etc. Another one is the recent technology, genome editing, which has the 3 method, CRISPR-Cas9, TALEN, and zinc finger nuclease, which is highly precise tool for modifying the specific DNA sequence to introduce the trait of interest like the drought tolerance, disease resistance, and improve nutritional contents of the crop. This precision tool is being utilized to edit the rapeseed and other crop rice, offering faster and a more targeted reduction of the anti-nutrient factor. These tech tools I will explain in the coming slides in details. Third one may be the RNA interference. This is also a genetic engineering technology in which the RNAi is used. In this technology, the lower or silence the expression of specific gene, which is not useful or which is uh, not useful to the plants, often used for improving the crop quality and the resistance. Fourth one will be the plant tissue culture. This technique is used for the micropropagation, rapid cloning, somatic embryogenesis, and generating pathogen-free plants, enabling the fast multiplication and the germplasm conservations. And the fifth one is the molecular marker. Use of molecular marker is important for the development of the different plant with gene of trait of interest like disease resistance, gene identifications, high proteins, and which significantly increase the or speed up the traditional breeding cycle as compared to the traditional and conventional breeding cycles. This thing will be explained in the coming slides. Genetic engineering in the plant is the introduction of specific fragment of DNA through genetic engineering tools. In this, we use a vector such as the Agrobacterium trinifications to introduce a foreign genes into the host plants, like the BT genes from the Bacillus thuringiensis into the plant, right? Brinjal, maize, rice. And another one is the biologic transformation. This is also another genetic engineering technology in which the gene gun is used instead of the agrobacterium vector. Third one is the electroporation. This is the high voltage electric power is used to deliver DNA into the host cells. Fourth one is the polyethylene glycol PEG mediated protoplast transformation. In this, the DNA is directly introduced into the protoplast of the plant. So limitation is success depend on many factors. Expected and unwanted reasons happens due to the random insertion of the target DNA, humans and environmental safety or ethical concern in these genetic engineering techniques. So in brief, the agrobacterium cell is used in which the TI plasmid is modified by inserting the foreign DNA. And then this recombinant TI plasmid is again transformed into the bacteria, agrobacterium bacteria. And this agrobacterium bacteria infect the host plant and transfer the tDNA, which carry the gene of interest. And the plant is generated through tissue culture, which has the gene of interest. So this is the basic flow chart of genetic engineering in the plants, conventional genetic engineering. So this is one good example that a japonica, a rice variety, which is white rice, was engineered with 3 genes taken from the daffodils and the Erwinia bacteria to produce or increase the beta carotenes. 2 genes was taken from the daffodil plant and 1 gene from the bacterium that induce the expression of the beta carotenes in the rice grains. And as you see the picture, the 2 rice grains is totally different. One is the golden color and another is the white. So golden color is because of the beta carotene contents. So this is known as the golden rice. This is achieved through genetic engineering. So another technology which is recently very popular and widely used was genome editing. This is a type of precision genetic engineering compared to the conventional genetic engineering. There is precision where in a specific fragments of DNA is inserted or deleted or modified or replaced in the genome of a living organisms. Genome editing is considered relatively a separate approach as compared to the conventional genetic engineering, and the crops that have been produced using such technique have been proposed to be subject to separate regulatory law than the genetic modified organisms. And this new technology offers higher degree of specificity in precision editing of DNA sequence. So in the genome editing, also known as the site-directed nuclease, the use of the site-directed nuclease enzyme, a specific type of protein to recognize and cut a specific site on a DNA sequence. After digesting or cutting the DNA, there are 2 ways to repair it. One is the non-homologous in-joinings. In this, an error-prone mechanism causing the point mutation during the repairing of double-stranded DNA break in the plants, and thereby inducing the artificial mutation. It may be deletion, insertion, or replacement of that particular DNA sequence. And the second one is the homology-directed repair. A homologous sequence is inserted to replace the target sequence. So there are 3 tools for genome editing. One is the zinc finger nuclease. Second one is the transcriptional activator light effector nuclease. And third one is the CRISPR-Cas systems. So zinc finger nuclease, this inhibits an artificial restriction enzyme that contain the zinc finger DNA binding domains and a separate DNA cleavage domains. There are 2 domains. One is the zinc finger DNA binding domain that will bind the DNA, and another is the separate DNA cleavage. That enzyme is another enzyme. These 2 proteins are located to the specific target of the DNA sequence. In this figure, FOKI is the enzymes, and the 9 nucleotide target size, right and the left, this is the binding site, DNA binding site. They will identify which side of the DNA they should bind. Once it is bind, the fork one enzyme will clip the DNA from both sides, and there is a double-stranded break. And this double-stranded break is repaired by the either non-homologous or homologous repairing mechanisms. But the limitation is that the zinc finger domain can be engineered or to target specific desired DNA sequence, and this enables zinc finger nucleus to target unique sequence within the complex genomes. And it is very complicated and technically challenging process of designing the construct and a low efficacy. Designing of using this technology is very complicated, and this very technically challenging. To conduct such type of experiment, people need very highly technically challenging experience. So second come the talents. Talent transcription activator light effector nucleus. It is similar to the zinc finger nucleases. Here also DNA binding domain will be there, and nucleus enzyme will be there, which have an affinity and a bind on the specific site to induce a mutation by insertion or deletion of a gene sequence. Double-stranded break in the target DNA, which is subsequently repaired by either of the above mechanism like NEISER, ISR, or HDR. However, compared to the zinc finger nucleus, it is more simplicity and a flexibility of design as well as their gene targeting efficiency. However, as compared to the CRISPR, these two above technology are complicated and need high skills. And this CRISPR-Cas technology is very simple and can be replicated or can be used in many ways. However, before going to the detail of this technology, this CRISPR-Cas system it can be expanded as clusterly, regularly interspaced short palindromic repeats Cas systems. The technology was taken from the bacteria. Usually, the virus infect the bacteria and hijack the bacterial cellular machinery to assemble their new viruses and thereby offal kill the bacteria. So, in order to defend these viruses, bacteria has developed one defensive mechanisms against this virus. So, this virus use the restriction endonucleases, which clip the viral DNA. How to cut the viral DNA? So, this is the CRISPR-Cas systems. And in short, it is a bacterial adaptive and inheritable immune systems against the virus. How the DNA is cleaved by the bacterial CRISPR-Cas systems. Here, in the natural process of the immunity system in bacteria, a single nucleus Cas proteins clip the invading the foreign DNA sequence of the infecting virus or plasmid, that foreign DNA to the bacteria into small segments. And then this small segment is interspersed as a palindromic repeats called the CRISPR array in the bacterial genomes, which will serve as a template for the CRISPR RNA infuser to defend against the infuser infection by the viruses. As you see in the figure, fragment is the DNA from the viruses. They have interspace in these locations. Then this trace RNA is transcribed as a RNA, and then this pre-cRNA is synthesized, and then they assemble as a pre-cRNA and a trace cRNA complex. This will find the complementary DNA from the foreign DNA, and then with the with the complex of the Cas9 complex, they will destruct the embedding DNA. So this Cris RNA then hybridized with a trans-activating RNA and form a dual complex and guided the Cas protein to detect, clip the foreign DNA in the subsequent infection of the virus, thereby protecting from the virus infections in the future. This novel technology has been derived from the adaptive immunity system of the gram-positive bacteria Staphylococcus pyrogens. There are two, however, in order to utilize this technology, we have two components. We require two components. So one is the single guided RNA. This will guide the Cas9 to the target DNA. So single guided RNA, in short sgRNA, is a short synthetic RNA which function as cRNA and trace cRNA dual complex of the bacteria by helping in recognition and binding of the Cas protein along with the 20 or 25 nucleotide unique sequence matching at target site in the upstream of the PAM motif, PAM, autospatial adjacent motif of the target genes. So PAM is required for the binding of the single guided RNA of the interest or genomes. So this single guided RNA has two functions. 50 regions, 20 base pair of the single guide RNA, thus base pair with the target DNA, and 30 part form a stem loop structure. As you see in the figure, that 50 nucleotide region is required. In that, 20 base pair is the binding with the target DNA, and 30 base pair is the formation of the stem loop structure. In that, the Cas enzyme will bind. Second is the enzymes, Cris-associated endonucleases or Cas proteins, for the cleavage the target site in the genome and inducing the desired deletion or insertion mutation at the target site. The most common target using this technology is to know out completely the function of the genes to produce non-alleles. Generally, this technology is used for the inhibit the gene expression or knock out the gene expression. That gene are not useful or not gene are negative effect to the yield or any other traits. So one is the gRNA, another is the Cas9. They will make complex, Cas-G9 complex. They will search the target DNA, bind it, and then they will cut the double-stranded DNA. And then the result will be repairing. In that, the same sequence may be written as, that will be the wild type. Another may be the insertion of the some nucleotide, or some other, it may be the deletion of the some nucleotide, or maybe the frameshift mutations. So all these complex is this is the brief. In that, there may be three techniques, site-directed nucleases techniques. First one is the small deletion or insertions, SDN ones, which generate the random small changes in the insertion deletion in the genome. So it does not require a repair template and does not introduce foreign genetic materials. In this case, the either may be the insertion or deletion one or two nucleotides. So it does not require any foreign DNA genetic materials. Second one is the specific modifications. In this, we use a small repair template to guide the cell to make specific or small changes like substitution or short sequence. BT gene is inserted to the plant genomes. In this, involve the insertion large DNA fragments or entirely new genes, foreign genes, into These are the DRR rice, also known as the Kamala, and the Pusa DST Rice 1, which was launched on May 4, 2025 by the Union Agriculture Minister Shri Shivraj Singh Chauhan. These varieties were developed by using this CRISPR-Cas technology by the Indian Institute of Rice Research, Hyderabad, and the Indian Rice Research Institute, New Delhi. DRR Rice 100 is developed from the Samba Musuri variety, in which the cytokinin oxidates two genes, which was knocked out. And because of this knocking out of these genes, the grain number of the per panicle was increased 20 days earlier, and it is found to be drought tolerance, less nitrogen, and as a result of this trait, it is 19% higher yield compared to the Norman Samba Musuri variety. Another variety known as the Pusa DST rice ones, which was developed from the MTU1010. This is also a mega variety of rice. And in this variety, one gene, DST genes, drought and the salinity tolerance gene was mutated or knocked out by the CRISPR-Cas technology. And because of this, the yield increase was ranged from 9.66 to 30.4% in the stress conditions, especially in the alkaline and saline soils. So these two variety was released last year by the our Honorable Union Agriculture Minister. And India is the first country to develop the genome-edited rice variety in the world. So another technique is the plant tissue culture. As a basic, plant tissue culture concept is based on the cell totipotentions, just like the stem cell in the animals, that ability to develop into whole organisms. So in this, an explant interorgan or intertissue is utilized to establish a whole plant in a sterile environment using the appropriate nutrients media. So here, as you see the figure, one part of the plant was taken and cultured it and developed into a fully developed plant. So another is the cell culture, is the cultivation of cell on a solid gel media or in a liquid medium, and known as the cell suspensions. Another one is the meristem culture, is a technique in which the apical meristems, lax vascular systems, are used as an explant to generate the disease-free plant. This is commonly used in the virus-free plants because meristem is considered as the virus-free plant, virus-free part. So if we culture the meristem, the virus-free plant can be developed. So these are the technique. The shoot tip is taken, and then surface sterilization, and then wash with distilled water, and then cut into small apical tips and put in the MS media and incubated in the regular photoperot and temperature, and then shoot is developed, followed by the rooting media for development of the root, and then transferred to the pot for hardenings and in the field conditions. Callus culture. Another culture is the callus culture, which involves inoculating an explant in a suitable mediums to differentiate and multiply, resulting in the formation of the callus, which can be induced to re-differentiate on an appropriate medium to develop embryos, then ultimately develop into a plant. In this, any part of the plant is induced for callus and then develop sooty callus, and then fragile callus, and then compact callus, and then embryo callus, and then complete plant is developed. Another is the molecular marker. Why molecular marker is important? Because molecular marker is used to select our gene of interest and it held in the plant breedings, whether we want to transfer the drought resistant genes or the insect disease resistant genes to the plant in very short generation. So molecular marker is a gene or DNA sequence with a known chromosome location controlling a particular gene of or interest. But Morphological marker is a visually distinguished quality like the seed structure, flower color, growth habit, and other important economic traits. But the limitation of the morphological marker is that polymorphism. Simple sequence repeats, single nucleotide polymorphism, and a cliff amplified polymorphic sequence marker. So, in brief, random amplified polymorphic marker was the first marker, DNA-based marker developed, and it was developed by the Williams et al. 1990 and the Weiss and the McLaren 1990s. In this is a PCR-based molecular marker technique that involved the amplification of the random fragments of DNA sequence using the PCR. We saw the difference in the pattern of the band amplifier, as you see in the figure, sample A and sample B. And then the 10 nucleotides, it employs a single primer, 10 nucleotides, for the random amplification under specific PCR conditions. As you see, the arrow is the primer and it is binding. So when you see in the electrophoresis, the A and the B, they are different bands. So the number of amplified fragments depends on the distribution and the number of the annealing sites for the genomes. The advantage of this marker is very quick and easy to assess. However, it is very low reproductivity, dominant type, and it is influenced by the DNA degradation relative to the regions with one or two primer, and it is very limited applications. Nowadays, it is not used. Another one is the star marker, sequence characterized amplified regions, is improved version of the RAPD marker. In this, the modification and conversion of the RAPD into a codominant marker and a more locus specific and reproducible SR marker is developed. As compared to the RAPD marker, this is more modify and a more better marker. As you see that the A and the B sample, there are three bands. One band is there. So this one band is used for designing specific primers. And then as you see, there is a more reproducible. This is the resistant fragment length polymorphisms, which is developed by the Boston et al. 1980s. This is more robust and more efficient compared to the RAPD. It was the first molecular marker technique and only marker system based on the hybridization, and which use restriction enzyme that cut the DNA fragment at specific site called the restriction site, resulting in different fragment of the variable lengths. As you see, for example, from the plant, we extract the DNA, and total DNA is isolated, which is digested with the restriction enzyme, either theta cutter or hexa cutter. And then this DNA fragment digested by the endonuclease enzyme is separated in the gel electrophoresis. And then this DNA is transferred to membranes. That also called the southern blotting. And then this membrane is hybridized with this DNA prof, specific DNA prof, using the radioisotope. And then this is recorded in by exposing to the X-ray film, and in that way, the bending pattern of the sample A and sample B is identified. The advantage of this technique, this marker, is the large range genome coverage and abundant in the genomes used across the species, and no need sequence information, highly reproducibility, and a co-dominance. But disadvantage is that it needs the radioisotope labeling, need good quality of the DNA in large amount, and it is very difficult to automate, more laborious as compared to the RAPD. Another improved version of the RFLP, that is the AFLP. It uses the restriction enzyme that cut the DNA, and the adapter are attached to the end of the fragments. The fragments are then amplified using the PCR, and their varying length can be then visualized on the gel or capillary-based platform. It's very sensitive for detecting the genetic polymorphism, but require large amount of DNA. So this is the combination of the RAPD, PCR-based, and hybridization methods. Another one is the simple sequence repeats. That is, simple sequence repeats are a simple sequence that arrange in tandem repeat of either mononucleotide, dinucleotide, trinucleotide, tetranucleotide, pentanucleotide, hexanucleotide, as they are highly abundant, polymorphic, and randomly dispersed throughout the nucleus, chloroplast, or mitochondrial genome of many species. As you see in the figure, the arrows are the repeats, either may be dinucleotide or trinucleotide. So one is having 10 to 60. So when we design the primer from the unique sequence, flanking sequence of this repeat, we will find different amplicon size. In the gel picture, one is the 9 CTG repeats, another is the 8 CTG repeats, and another third one is the 7 CTG repeats. So number of repeats, based on the number of repeats, the amplicon size also different. In that way, we can identify the polymorphic. Another one is the Cliff amplified polymorphic sequence marker. So it is a combination of the PCR and RFLP technique, where a very small amount of DNA is required for the PCR analysis to show the polymorphisms, as you see in the figure. And the K marker we are successfully employed in many perennial plants due to availability of their nucleotide sequence in the public domains. And another is the SNP marker, single nucleotide polymorphisms. So it describes a single base difference between the two DNA sequence. As you see, the one is the female plant, another is the main plants. Here we find one SNP polymorphism, either C or T. So this type of polymorphism is called the single SNP marker. So this can be identified by the sequencing or some CAP marker also. So now, by using all these biotechnological tools, the enhancement of the livestock nutrition or health can be achieved by implementing a balanced diet featuring high quality forages, energy sources, protein supplement to improve the productivity, growth, and immunity. One may be the antioxidants. By incorporating the natural antioxidant reduces the oxidation in feed, keeping it nutritious for longer. Another one is the disease-resistant crop. If we develop disease-resistant crop using the genetic engineering like BT maize, which will offer the increased resistance to the pests and disease, and ensuring a more consistent toxin-reduced feed supply. So there will be no fungicide, no insecticide. So that fodder will be good for the livestock. Another one is the enhanced digestibility of the fibers. So that biotechnology can alter the lignin content in a forest crop like alfalfa, increasing their digestibility and improving the energy intake for the ruminants. Fourth one would be the functional feed component. The recombinant DNA technology can be applied to introduce the genes that produce the beneficial substance like the industrial enzyme cellulase directly within the fodder, aiding in the breakdown of the complex carbohydrates. The fifth one will be the immunomodulation biofeeds. Plant can be engineered to express the vaccine or antibiotics, offering a cost-effective way to protect the livestock against the disease through their diets. Six may be the improved amino acid profilings to increase the nutritional value of the fodder. Genetic engineering can be used to enhance the nutritional value of the feed by increasing the level of the essential amino acids like the lysine, methionine, threonines in the grain crop, reducing the need for costly synthetic supplements. One is the low phytic grains. The high phytic reduce the bioavailability of the iron, zinc. So food should be low phytic. So genetic modification has to be developed of soybean or maize with low phytic levels, which increase the bioavailability of the phosphorus and other minerals for monogastric animals, poultry and pigs. And another one is the protein and energy optimizations. So utilizing high quality fodder like alfalfa and protein rich feeds, jack beans example, alongside the corn and molasses balance the energy and the proteins of the livestock. Last one may be the reduce anti-nutritional factor. This technology can reduce the anti-nutritional factor like the tannin, glucosinoids, and cyanogens in the legume-based foods by modifying the genes, manipulating the enzymes. So example include the development of the oil-shaped rare cultivar with low erucic acid and the glucosinolates. So I will listed some example. This is the published in the Journal of Agricultural and Food Chemistry. In this, the flex plant was engineered to increase their antioxidant capacity and improve the oil composition and the stability. As you see, the transgenic plants, in this, the transgenic plants produce more 20 to 45% polyunsaturated fatty acid than the control. And that polyunsaturated fatty acid are merely alpha-linoleic acids, thus increasing the antioxidant potential of the flex plant. This will be suitable and benefit to the humans and animals' dietary consumptions. Second, maybe the engineering the plant with increased level of the antioxidant chlorogenic acids. So in this also they have modified genes in the in this one tomato. Here the overexpression of the high QT genes in the tomato causes plant to accumulate higher level of CGA, chlorogenic acids, with no side effect on the level of other soluble phenolic. And it saw the improved antioxidant capacity and the resistance to infection by the bacterial pathogens. So these are the example of the enhanced antioxidants in the plants by the genetic engineering or biotechnological tools. Another one also published in the scientific reports that they increase the protein content and the quality of the protein by overexpressing the artificially synthetic fusion proteins with high lysine and the threonine constituent in the rice plants. This was published in 2016. And another is they change the or limit the expression of the one genes, IPA genes. And by modifying these genes through the genetic engineering, they have lowered the phytic content in the maize. So this will be also useful for as a fodder to the animals. Here the precise DNA editing to simultaneously improve the nutritional and avoid phenotypic effect also. And phytic acid is considered as an anti-nutritionals in animals, humans also. And another one is the development and evaluation of low phytic acid soybean by the siRNA triggers seed-specific silencing of the inositol polyphosphate 635 kinase genes. This is done in the soybeans by the silencing of these genes to reduce the phytic contents in these soybeans, which was published in Frontier in Plant Science 2018. And another is the development of the low phytic rice by RNAi, one of the technique in biotechnology that seed-specific silencing of the inositol 1, 3, 4, 5, 6 pentakey phosphate 2 kinase genes, IPK1 genes. This gene was mutated in the rice by the RNAi and it reduced the phytic content in the rice grains. So in this way, there are so many GM crop which is used for the livestock feeds. In alfalfa, herbicide tolerance, modified product quality, there are a number of 5 events, 5 GM crop. In apple, non-brownings, 3 events, Argentine canola, herbicide tolerance, modified product, pollination control system, 37, beans, disease, viral disease resistance, chicory, herbicide tolerance, pollination control systems, cotton, insect resistance, herbicide tolerance, coffee, insect resistance, creeping bane grasses, herbicide tolerance, eucalyptus volumetric wood increases, flax herbicide tolerance, maize, papaya, disease resistant, plums, Polish canola, potato, rice, insect resistant, herbicide tolerance, safflower, soybean, in soybean modified product quality, herbicide tolerant, insect resistant, alter the growth in the yields, squash, sugar beet, cotton, tobacco, tomato, wheat, etc. This is the this taken from the ISAAA GM approval database. So this all these as a conclusion, these are the technique that can be useful for the enhancing the nutrition and thereby the health of the livestock. So nutrition strategy targeting gut health, reduce the inflammation, and improve the animal ability to combat diseases, high productivity, increase the milk production, better weight gain, high quality meats. And it also improve the fertility, proper nutrition, particularly which mineral interact. And this, by all these, it reduced the veterinary expenses, like healthy, well-nourished livestock are less susceptible to diseases. I conclude here. Thank you. Any comments, please?
Thank you so much, sir, for enlightening us. So now the session is open for question answers. So participants may post their queries in the chat box.
Thank you, sir.
We will be waiting for another 2 to 3 minutes for the queries of participants. So if there is no query, so I would like to thanks to you, sir, for delivering such an informative session for our participants. Thank you so much, sir.
Thank you, Nisha. Thank you.
Thank you, sir.
Thank you.
So with this, we are concluding the session 2 and also the day 9. So on behalf of the organizing committee, I would like to express our sincere gratitude to all the dignitaries, our speakers and participants. Thank you everyone for your active participation in today's technical sessions. With this, we conclude the day 9 of 10 days international training camp workshop on advances and innovation in veterinary science for viable livestock practices. We look forward to your presence tomorrow for another enriching session with our distinguished speakers and experts. Thank you once again. Have a wonderful evening. Jai Hind to all. Thank you.